And then what happened was a major event happened that changed the whole course of human history. That was correct, Betsy. It is a Human Genome Project, right? It was a joint British and an American effort. It was a race who will complete first. It was a race just not whether they have to take drugs, but whether they can actually produce them. And it was all the Human Genome Project was sequenced, right? So what happened was this ended up to be the biggest breakthrough in the field of informatics. And that has caused the birth of many, 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 many disciplines, right? And so we are now in a whole data tsunami, right? Big data. This is where it actually started, but it didn't end there. We are still doing a lot of data, right? And that's why we are all in a luxurious state that you can actually do projects, you can actually discover things yourself. It may seem odd, but each one of you can actually find something new because there's so much data that we still need a lot of people to actually look at this data. So now at the conclusion of the Human Genome Project, what happened? It actually created a whole new field of science and medicine called the genomics, right? And now it is completely a whole new field of genomics, virtual genomics. Then what happened was the human genome was sequenced, and then that it didn't end there. Then people started going crazy and started sequencing more and more genomes, right? They started sequencing, and at the end of the human genome, we had 3.2 billion base pairs of the human genome, right? And people started sequencing more and more genomes of Drosophila, rat, and chicken, and mouse, and all of those things. So when you think that we are the most important people on this planet, right? And we are all trying to solve diseases, we are all trying to cure diseases. When once the human genome, when they declared that it was sequenced, why do you think it is important to actually sequence the genomes of rats and mouse and chicken? Why do we care? Why do you think we have to sequence other genomes? Why did people do that? Any ideas, any guesses? Yes, comparison, right? That's right. So you want to compare because you know what? We cannot do, and to study evolution, that is perfect, and to study evolution, but you want to compare comparative genomics, right? For example, if I want to find a cure for a disease, even for COVID, lab test, correct. So even if, for example, COVID-19, right, we are trying to create like what, 20 different groups in the world are creating vaccines, trying to do vaccines. You cannot do all of that in humans, right? To do all the testing, we have to first use what are called the model organisms. We want to test that before we actually test it in the human, right? So that's why we have mouse, rats, and chickens, and everything else was sequenced so that we can do comparative genomics of what is different between humans and them for various research purposes, right? To create a disease model. And so now, but what happened is because we have sequenced so many genomes now and so many everything else, and we have so many proteomes right now, it also comes with a lot of different challenges, right? The challenges that we are actually facing today is the number of sequences is exploding. We have 106 billion nucleotides. We have more than 6 million data from environmental sequences alone. I'm not even going to talk about this unless one of you is interested. I can talk about this later. And then we have lots of data in UniProt knowledge base, which we're going to be talking about, and you will have lots of hands-on experience with UniProt. And then from the sequences, we have domains, we have three-dimensional structures, we have protein families, we have clusters of orthologous groups, and all of those things. This is just, we are just talking about proteins and DNA. I'm not even talking about other data sources like medical, electronic health records, medical records, and any of those things. That's going to be beyond the scope for this lecture. So I'm not even going to talk about those things, right? And now, how are we going to deal with all of this data explosion?